welcome everybody to the webinar between the National Biofilms Innovation Centre and Cosmetics Cluster UK. So in this new world where we all talk to each other via Zoom, then it's a real pleasure to welcome over 100 people to this webinar. So thank you very much for your interest. So the topic of the webinar is the science behind the skin microbiome and biofilms, evidence and claims. The reason that we're holding this webinar is because of the increasing interest in the skin microbiome in the cosmetics and personal care sector. And we have some really good contacts, not least our own Katerina, who are very knowledgeable about the skin microbiome, and also about what you'll hear today is about examples of the way in which the skin microbiome is being marketed, examples of the growth in the market sector, all the intricacies of the regulatory position that companies are going to be facing as they start to get into this sector, and some of the interesting science, and perhaps more particularly some of the things we don't know about the science. So if we go to the next slide, you'll be able to see the agenda for our meeting. So this is me giving a short introduction. The first talk is going to be between Katerina Steventon from NBIC and Kristin Neumann from MyMicrobiome, followed by Olivia Santoni, who's going to talk about the regulatory landscape. And Olivia runs her own regulatory company, Bloom Regulatory. This is going to be followed by a talk from Dr. Sudeep Basu, who's from Frost & Sullivan, who, as many of you know, will give us an information on the market and the sector and growth opportunities. And then we'll wrap up with a question and answer session to the speakers and to anybody else. And for those who are interested, I can also share some information about the cosmetics cluster, just to tell you a little bit about who we are, what we're doing. So without further ado, I'd like to introduce Katerina and Kristin to give their presentation, Skin Microbiome versus Biofilms, a New Approach to Formulation. Thank you.
Thank you.
And perhaps whoever's host can mute everybody while the speakers are giving their talk. And just to say, we are not recording, we are recording this. So participants don't need to record it themselves, and we will make the recording available afterwards when we will send you a form to give us some feedback. So thank you. So over to you, Katerina.
Thank you. Kristin, can you see the slides?
Yes.
The slides, lovely.
Okay, so I'll start. So thank you, Katerina and the National Biofilms Innovation Center for inviting me to this webinar and for letting me talk about our work and about the skin microbiome. And next slide, please. Our talk, Katerina's and my talk, is going to be about two different approaches concerning the skin microbiome, and the skin microbiome in health and the skin microbiome in disease, which would include biofilm formation on skin. So my part of the talk will be the testing of skin microbiome friendly formulations on healthy skin, and the aim of how to maintain a healthy skin microbiome. And Katerina?
So I help innovative companies with new research, with translational research in biofilms in personal care. And I will discuss today biofilm features in acne, which is a chronic inflammatory skin condition. And through the talk, I will remind you that formulating new personal care products should really take into consideration both the skin microbiome friendly and anti-biofilm approaches.
Yes, so that is how we want to close our talk then, is that we have to consider that the skin microbiome research is still in its infancy, and we keep learning, and we strongly advocate to advance the fundamental research on both sides, on the healthy skin microbiome, but also on the biofilm formation. And with this, I'll start with the presentation of our work. the microbiome friendly seal. Please, next one. And just first some background about the skin microbiome science and the research and what we're looking at the moment. And as we said, it's still in its infancy, but it's just as complex as the gut microbiome. And that's why a lot of research is needed to fully understand. And we are now understanding a little bit, but it's already starting with how do we look at the skin and how do we analyze the skin microbiome. And it's already starting with simple things like the sampling techniques, which already reveal different results. So most analysis done by swabs, and it would be better to have like stripes, or even better to have biopsy, but who would like to have a biopsy to get their microbiome analyzed? But to have a whole picture, you have to really think about better sampling techniques. And then we come to the next step, which is the analyzing method. Most research is done via 16S rRNA, which is only looking into bacteria and not even down to the strain level. But a whole genome sequencing would also take into account the fungi and also viruses which live in our microbiome, and it would go down to the strain level. And then in the next step, it is important to which database are these sequences being compared. So depending on which database is being used, you will have different results. So that is already something we have to consider always when we're looking at data about the skin microbiome. What we know about the microbiome of the skin is that we have different body sites, and the diversity between those body sites, as is explained in this picture, very nice. So I think everyone knows that one. It's higher between the different body sites than the diversity is between the individual persons. And we also know that the deeper we go into the skin layers, the diversity decreases. The skin microbiome is drawn by genetics, diet, lifestyle. It depends on if you have pets at home, your living area, if you're living in the city or in the countryside, and of course your hygiene behavior or medication you take. And we have a core microbiome, which is the deeper skin layers, and the transient microbiome, which you would probably, which is on the surface of the skin, and you would take like that if you make a swab of your skin. And the main genera that are found in the microbiome, in the skin microbiome, is Corynebacteria, Cutibacteria, Staphylococci, and Malassezia. Next slide, please. And this is just an addition. So also the pH and the temperature is varying a lot between the different skin sites, and that is why different environments flourish on the different skin sites, and that is harboring and making different bacteria and fungi flourish on those different body sites. So what is the microbiome actually doing with the skin? We know that it's of tremendous importance for our skin's health, and it's fulfilling a lot of tasks. For example, it's fighting off pathogenic bacteria when releasing AMPs itself, but it's also stimulating the skin's immune system to release AMPs. It is, for example, it's also influencing the pH and therefore making the environment harsh for pathogenic bacteria such as Staph aureus. It controls the immune system and it stimulates the immune system. So now the big question is, of course, and which every cosmetic brand would like to have, is how to improve the microbiome. And that is something that is really hard to tell, because at the moment, the best one can do is to just leave it alone if it's a healthy microbiome. For example, just one example is Staphylococcus epidermidis, which is a healthy, it's a commensal bacterium, but as soon as you might add some more of it, you create an imbalance of your microbiome, and it's not commensal anymore, but it might become pathogenic because it leads to an imbalance of your microbiome. Yes, and then, but also the genetic predisposition and external factors can lead to skin issues such as atopic dermatitis, rosaris, dandruff, dry skin, but Katerina will come to that later. Next one, please. So what are we facing at the cosmetics market at the moment? So we all know that if we use a lot of hygiene product and put a lot of products on our skin, it will in some way modify our skin microbiome. And of course, at the moment in this current situation, we need to wash our hands a lot and also disinfect it, but it also will result in skin irritation and changes within the microbiome. So we have to always consider what is better for our health. Do we really need to disinfect as much and then be more prone to skin irritations? The future of cosmetics will not further exist without the consideration of the microbiome, so that is for sure. So we have to take into account, or have to have a deeper look at preservatives, alcohol, plant extracts, surfactants, and but also antibacterials. And I have to say, I know a lot of companies that are working with us who do already a very great job in that, to use mild preservatives and less ingredients. But also on the other hand, there's a lot of brands on the market that are using the microbiome claim for marketing purposes without really showing scientific data behind it. And a lot of them use the word post, or prebiotics for the skin, which sounds nice. But in most cases, I don't say it's like there's post and probiotics, which show a benefit, but in most cases, it's only marketing. So in most cases, it's lactobacilli used, and they're not even alive. And they're claiming to be microbiome balancing, improving, gentle, friendly, or supportive. But there's no clear regulation behind that or definition. So that is something that we're working on. And that is why we created that microbiome friendly seal. Please, next slide. So we were developing a seal called microbiome friendly, which would test cosmetic products on their influence on the skin microbiome, or the typical set of bacteria microbiome. In the current cosmetics market, and we are also working in a research group. Can you go back to the presentation one? So we're also being part of a group of the CCUK microbiome research group, which is trying or is setting definitions in the microbiome, skin microbiome field. We're creating definitions for the microbiome, microbiota, pro and postbiotics, symbiotics, dysbiosis, and others. And the principles of the group members is that we all have to be scientifically accurate. Can you go back to the presentation mode?
Yeah, can you not see presentation?
No, it's I see the PowerPoint.
I'm sorry. Okay.
You just need to put it back on. That's it.
Yeah. So okay, now we can go to the next slide. And yes, so we do acknowledge the uncertainty around the skin microbiome science, and we also always would adapt if something new comes up. And the group members are free to join, but they always would have to agree to the defining principles. And of course, we're facing the questions: how do we transfer definitions from the field of nutrition and health to cosmetics, which is kind of tricky because the definitions in health do not really, like, it's tricky in the cosmetics to have a health claim. You should not have that, but you should, on the other hand, somehow show that you have a benefit, you create at least a benefit, and that has to be shown in some way. So, next please. So some insights about the testing we are doing. We developed different standards for the different body areas. Also, for example, for infant skin or the vaginal tract, which is very defined. And we're looking at the key players of each different body site and like different bacteria and fungi, and we investigate how the product influences the growth and the vitality and the balance between those bacteria. And all of them are culturable, so that is how it's in vitro. Please, next one. So we're looking at the diversity of the key players in one co-culture. So that is because we want to also take into account the interplay between the bacteria and fungi in the co-culture in the presence of the product. But we also look at each bacterium and fungus individually in a vitality test. We're having a balance test where we have two different bacteria, one is pathogenic and the other is commensal, and we want to see if the product favors the growth of one specific bacterium in that co-culture. And actually at the beginning of all tests, we perform a microbial quality to make sure that there's no contamination in the product. So next, please. We are already working with numerous big players from the cosmetics industry, and there's a lot more. This is just an excerpt here. And yeah, we're very proud because the companies are really keen to develop microbiome-friendly products. So in case some products don't pass the test, they would reformulate their products and retest until it's fine. Yeah, next, please. This is just an example of how one product, which is not microbiome friendly, might look like. On the left-hand side, you see the control. This is, I think, Staphylococcus capitis, and without, as a control, without any product. And then on the right-hand side, you see how many bacteria are being killed by the product. Next one. Well, I'm coming to the conclusion of my part. So what we know so far is that the skin microbiome is science or research is still in its infancy, and the claims that one can make is actually very limited, because the best one can do to a healthy microbiome is not to touch it and to leave it alone. But there's a lot of claims out there already and no independent control body. That's why we developed a microbiome friendly certification, and it's the world's first and only independent control body. And we're always looking at the science and the development of the science, and we always have science-backed claims only. And we're working together with skin microbiome groups, where the goal is that we set a momentum in the cosmetics industry with true scientifically valid definitions. And with this, I would like to give the word to Katerina.
Thank you, Kristin. I'll just ask everybody to mute themselves, please. So I'll explore the relevance of the biofilms modality in personal care. And in our publication in Cosmetics and Toiletries earlier this year, we have postulated a definition. So a biofilm is the predominant way the microbiome establishes itself in any ecological niche, including the skin. So just again, back to definition. So if microbiome is the total genetic composition of all species on the skin, then the biofilm is the biological environment in which these cells grow, evolve, adapt to their surroundings. And on the right-hand side, you can see the biofilm life cycle, which starts with reversible attachment, continues with irreversible attachments of the cells and the growth of the biofilm, then maturation and dispersal. So again, biofilms could be good or bad. So good, positive in the gut health, negative, for example, in wound healing. Biofilm as such is well-characterized mode of growth, both of the single or multi-species populations. They become a community and have cooperative behaviors, which are very different to individual bacteria that live as single cells. Once bacteria become aggregated together, they protect themselves by producing slimy extracellular matrix, so-called EPS, which is really rich in biopolymers, and they become more virulent and also more tolerant to antimicrobials. We've seen the map, and I'll just quote Tony Rawlings and look at his work for DSM. So stratum corneum is desquamating, and therefore the substrate for attachment of bacteria is counteracted by the cell shedding. DSM in their blog, The Secret Life of Skin, also quote that new approaches are needed for skin research due to the structural complexity of the skin, as well as small microbial concentrations. So looking at the biofilms, skin microbiome can display phenotypes with biofilm features in dysbiosis, and that is even prior to the biofilm formation and laying down the EPS. And biofilms can be present on the body, primarily in disease or in sebaceous or moist skin areas. So the skin conditions like acne, and dermatitis, scalp health in terms of dandruff, the intertriginous niches like axilla or feminine health or athlete foot. I will explore acne today, but we have written about the other areas, for example, the feminine health in our blog. So NBIC, the National Biofilms Innovation Center, how we work with personal care industry. First, we facilitate partnerships for translational research in biofilms. We have a consortium of leading academics with research excellence and capability. And we engage in a dialogue with the industry so that we can better understand the unmet and sometimes even unarticulated consumer needs. We identify the solution that fit within the cosmetics regulatory pathway and translate the research into either industry know-how or IP and eventually consumer claims. We operate within 4 interventional themes: prevent, detect, manage, and engineer biofilms. And those themes which are relevant to the personal care industry are detect, so accurate, quantitative, and actionable detection of biofilms, which is in real time, quick, and in situ. Or the management of biofilms, which is about killing, removing, or controlling established biofilms, targeted disruption, understanding the quorum sensing, and then translation of these anti-biofilm strategies into either in vitro models or bioavailability testing. So we are an innovation hub, have research and industry partners, and we believe that true drivers of innovation are, first of all, excellence in science, but also its fast translation from the academia to the industry. So a few of our industry partners, companies large and small, so the ingredient manufacturers, the finished product manufacturers, testing houses or SMEs. And within our academic expertise, some of you are joining us. So I'll start with University of Manchester, expertise in skin microbiome and epidermal lipids. Universities of Surrey, Sheffield, and Manchester, again, having different in vitro models with specific microenvironments. University of Edinburgh has expertise in soft matter physics, guiding the formulations and reducing biofilm formation. Ulster University, expertise in probiotic strains and engraftment in acne follicles. Matt Hardman is joining us today. So University of Hull, skin microbiome in health and aging. Glasgow, expertise in feminine health and microbiome. Aberdeen, looking at fungi and hosts and microbiome interplay. And also the underarm health, University of York, and again, Edinburgh. So just a few handpicked from our 52 universities. So when we sit down with our industry partners to explore their industry needs and opportunities, these are the questions they usually ask. So really explaining where biofilms fit within skin microbiome. Looking at clinically relevant models and sampling. So what are the appropriate sampling, sequencing, and quantification techniques? Can in vitro modeling provide leads for clinical implications? Diagnostics pairs now with personalization of our microbiome. So again, the quick, relevant, standardized, accessible, point of care, simple and inexpensive techniques are much needed. Biomarkers, which are definitive for biofilms, looking to characterize, detect the dysbiotic shift from healthy to unhealthy state before it happens. Looking at detecting and confirming the presence of biofilms, again, with standardized and reproducible methodologies which are acceptable to the regulators. So I'll just speak very briefly about biofilms in acne. It is all about understanding the follicular environment. Acne is a disorder of sebaceous glands and very common skin condition affecting just under 10% of global population. And Staphylococcus epidermidis, Kristin mentioned before, and Cutibacterium acnes, they are known to contribute to acne, but they are also commensals promoting skin health. So the four factors in onset of acne: increased sebum production and hyperkeratinization, hormonal changes, and C. acnes historically was considered the culprit. Yet now we know that equally present, it is equally present in follicles of acne and healthy skin. And with the direct visualization of C. acnes in skin biopsies, we can see highly complex macrocolonies or biofilms in acne follicles, and we understand that it's not the species but the strains. Therefore, chronic inflammation comes probably due to the loss of the diversity of the skin microbiome and also activation of the local immune response. And I can share with you the presentation for the references later. So picture speaks 1,000 words. So just again, it is all about relative abundance of C. acne strains. And this is a publication published in Nature in 2016, looking at, on the left-hand side, normal relative abundance of dominant C. acne strains in healthy skin. So you can see 10 different strains. And then B, the dysbiotic shift, so upregulation of some and decrease of the RT3 and 6. And in the C picture, persistent nature of acne is that due to the pockets of biofilm forming strains. So again, to understand the disease clinically, we need to be able to model the follicular environment, and just a few examples of in vitro models. So in 2007, a Belgian group has looked at biofilm forming ability of C. acne strains in vitro, resistance to anti-acne agents, and production of virulence factors. And in 2018, they developed the model and published further, looking at artificial sebum model, which is representative of the follicle, inoculated with C. acnes clinical strains from both normal and acneic skin to again study growth and biofilm formation and virulence. And research from last year looking at the acneic and non-acneic strains of C. acnes as they adapt to the environmental niches. So Olivia will speak more about claims, but we know that anti-acne claims are limited by the cosmetics regulations. So they are around helping prevent skin breakdowns, removing excess sebum, dirt, impurities in the pores, creating a healthy, clear complexion. And the ASA, the Advertising Standard Agency, reminds everybody that efficacy claims need to have robust clinical evidence. And consumers now demand more of natural therapies, technologies, which are well tolerated but have limited efficacy. So these could be plant extracts, a tea tree oil, barberry green tea, small molecules like niacinamide, or probiotics, which can manipulate the host microbiome. And C. acnes and S. epidermidis have been trialed in skincare. It very much depends on the dose and on the engraftment. So overall, the target would be enhancing skin microbial diversity. The detail of what and how is not yet known. And the first step for research is to understand the microbiome signatures and biofilm formation in health and disease. So in my conclusion, looking at both of these approaches, so skin microbiome friendly and anti-biofilm, is important when formulating new products. Our understanding of healthy skin microbiome is very much in its infancy, and we advocate advancing fundamental research in both of these areas and keeping the dialogue alive, understanding both of these modalities. And please come to talk to us at NBIC. Our academics are world-leading experts in skin microbiome and biofilm research. Thank you. So Olivia, I believe it would be you now, continuing with your talk.
Katerina, apparently they say host disable participant screen sharing. Can you try to make me a co-host again?
Yes, of course. Sorry, hold on. Here you go. Can you share now?
Check. Yes, it works now. Perfect. Thank you. So let me just open that. Good. Oops. So yes, thank you, Katerina and Kristin, for the presentation. I think it's a good introduction, as you say, about the science behind, and I think the regulatory landscape, I think we are in the same stage, is that there is still a lot of evolution to be done. It's a big topic. I think we were a little bit like we were in nano a few years ago, so microbiome is starting to be very interesting in the regulatory landscape. And what I will try to do today is maybe explain some of the challenges, where we are heading, and what are the issues of the regulators, because yeah, of course, we are all working with industry, with development, but at the end of the day, we need to understand where the regulator come from, what are the challenges, and is the current regulatory landscape sufficient enough to ensure that the products are safe, efficient, but also provide, I would say, some sort of a classification of a cosmetic as well. So just a little bit of word about our company. So Bloom Regulatory, we are offering a regulatory consultancy. We are a relatively new company, 2019. And what we tend to do is really looking at this type of topic when it's more of a global expertise approach on complex issue, and looking not only at the regulatory landscape, but the legal landscape, supply chain, and trying to provide some solution as well. So we, of course, have customers that want to make microbiome claims, not only for Europe, but also for the US, for Japan, for Korea. So we have to try to understand the regulatory landscape and advising them on that. I think let's start from really the basic. There is no international guidelines in terms of regulation, the terminology applicable to cosmetic ingredient that work with the skin microbiome. But I have to be honest with you, there is a big interest about this topic, and in particularly from regulators. They are meeting to discuss this topic. There is a huge interest because I think it's the development, and we can see a lot of discussion happening to try, I would say, which is a good thing, because I think what the objective would be with regulators is to try to get a global approach to it. So not having so much difference between Europe, US, Canada, but more a sort of a global approach, so to make sure the consumer is protected. And I would say, well, this is something that was already touched by Kristin and Katerina, but some of the definition about probiotic with the viable microorganisms that is added to a cosmetic product, the prebiotic when we're talking about a non-viable ingredient, and the postbiotic, again, non-viable ingredient comprised of inactive microorganisms or soluble factor released by live or inactive microorganisms. And all of that are currently being discussed by the industry, by companies, to be added in cosmetic product, and at the end of the day to provide the benefits to this product and claiming such benefits. So I was thinking, well, what does regulator think about microbiome? And I think the US FDA has been one of the most talkative about that. They were highlighting their issue with microbiome. There was an FDA conference about it, specifically on the cosmetics, because of course, microbiome, there is also other issue with the pharmaceutical or the food area, but on the cosmetic, the US FDA has been quite talkative about that. And if we look at some of the questions that was raised, I would say there is specific, I would say, key criteria. So they're looking at the efficacy issue. Are probiotics still alive in the cosmetics that are preservatives? The safety aspect also, does the plant of a probiotic have an effect on product safety? Or is there an effect on product quality? So the quality aspect. Is there specific consideration for manufacturing cosmetic containing probiotic? How can cosmetic containing live microorganisms meet existing microbial contamination limit? But what about the shelf life as well, the storage? And also we are seeing, in addition to the safety, quality and efficacy aspect, the borderline aspect. And I think this is where authorities are focusing at the moment. Katerina, you mentioned about acne, you mentioned about broken skin, eczema, psoriasis, atopic dermatitis. And this is, I think, where the authority is concerned in a lot of countries to say, well, you are adding this ingredient, but what is the benefit of it? Are we starting to say it's a medicine by presentation? And I think from an authority, you need to understand that it's not just because you are going to claim a medicinal claim that they are not happy with, that the consumer is going to be misled, but they also think that they may not get the right treatment if they actually have an adverse condition, if they choose a cosmetic. So we could see a lot of discussion or a lot of enforcement, I would say, happening more on the borderline issue than the rest of the aspects that are declared here from the US FDA. So now changing region, but also I think some of the principles here will apply globally. In the EU, we don't have specific regulation of requirement regarding cosmetic product or ingredient intended to work specifically with the skin microbiome or using some probiotic, prebiotic or postbiotic. But that doesn't mean it's not regulated. That means we didn't think it was necessary to specify that at the time. Maybe there will be evolution, but at the moment, that means we have to refer to the current regulatory landscape. So the cosmetic regulation, which deal with products and ingredients, but also REACH and CAP regulation about ingredient in this product. And we have also specific directive in the EU about work exposure as well. I would say there is no country in the world that have a specific regulation on microbiome cosmetic. So we have the same issue in Brazil, Canada, Japan, USA, Israel, South Africa. Other countries doesn't have a specific regulation for that. So you will have to look at the general regulation that governing cosmetic product. So this is, again, this will go, I think, very well with what Kristin was mentioning, is the fact that actually microbiome cosmetic or cosmetic with probiotic, prebiotic or postbiotic tend to apply for the full sector of cosmetic, not just skincare. We always think a lot about skincare, but we can see that in oral care as well, leave-on, rinse-off, antiperspirant and deodorant. Some of the prebiotic ingredients here, we can see it in cleanser, hair care as well. And the same with the postbiotic ingredients. We can even see them in makeup now, in a primer, foundation, mascara. So I think it's important to go beyond skincare, which I think is what we think about when we think about microbiome, but to understand that it is a science that go beyond just the normal moisturizer that we think about, or the cleanser. But in addition to add a specific ingredient that are either probiotic, prebiotic, or postbiotic, it's true that what we are seeing on the market, and again, is go totally in sync with what has been presented before, is more the not harming the microbiome. So microbiome friendly, so it's more how do you formulate a product that it will not have a negative impact on the microbiome. So we be a more gentle formula. As we mentioned, there is now a big appetite for the consumer to go for natural ingredients. So this is a way also to develop product with the microbiome in your development. But of course, we need to be careful about what we claim, if it is indeed what we are developing. And so I think here, I would say the clarification of the claim is very important. So really, when we looked at the regulatory landscape, I would say we have to do it by step. The first step is that can we use such ingredient in a cosmetic product? Then what are the safety considerations that we are seeing across the globe? Then the classification of the product, I would say focus on that one, because this is where we are seeing the most enforcement at the moment. And then the claim substantiation, as we mentioned, about making sure that what you say on the pack actually is true, and you have robust data for that. So if we look first at whether or not we are allowed to use such ingredient in cosmetic product, I would say, let's go on the EU side for the moment. We have a very, I would say, clear definition of cosmetic product in the EU cosmetic regulation, which refer to substance and mixture. That doesn't mean you can only have substance and mixture. Sometimes we are adding article in cosmetic product. We could see that, for instance, with nail polish with the little balls inside to make sure it's separated when you shake it. So it's just, it could go further than substance and mixture. Then we've got a definition of substances and mixture in the regulation. So substance meaning a chemical element and its compound in a natural state or obtained by any manufacturing process, including any additive necessary to preserve its stability, and any impurity deriving from the process used, but excluding any solvent which may be separated without affecting the stability of the substance or changing its composition. So here, there was a lot of thinking about that. Well, can a prebiotics and a postbiotics be considered as a substance? And I think here we are relatively, I would say, confident that it could fit the definition from a mineral state, from natural state. So I think in that one, we could see that it is, it could be considered as a substance. And as you know, at least in the European regulation, you can use anything you want as long as it is safe. So it doesn't need to be specifically approved except for certain type of ingredient like UV filter, preservative or colors, but the rest it's more about using ingredients that are not banned and are safe. When we look at probiotic, there is a little bit more, I would say, legal limbo here, because our definition of cosmetic doesn't mention viable microorganisms in the definition of substance. So this is really, again, how do you read the regulation? Does everything needs to be specified? Or actually when something is not mentioned, that for me it is acceptable. And I think, again, here there is no consistency. We look at the medicinal regulation, viable microorganisms are specifically detailed in the substance definition, but in food it is not. And we know that we have probiotic in food or in food supplement. So here again, I think we've got enough argument to say, well, actually, although it's not specified as such, and it's still a bit of gray legally speaking, at the end of the day, some company will take the position that it is sufficient to say that it is not banned and I can use it. So now that we are a little bit more clear about the ingredient composition, the safety consideration, I'm going away from the EU here. I'm going maybe more in countries that are more, I would say, focusing on the issue of microbiology. The US, we know they do a lot of tests. They have very strong regulation about microorganisms beyond the acceptable limit. They have their specific limit in the US and they do test. And if you are not complying to that, they consider it as a cosmetic that is adulterated, not compliant. Canada is the same. We required through a certain limit in terms of the microbiological and refer to the ISO standard. The Japanese standard for cosmetic, here again, we are talking about ingredient of cosmetic, including an impurity, contain therein shall not contain anything that may cause infection or that otherwise makes the use of the cosmetic a potential hazard. So here again, we are looking at the microbiology and the fact that it needs to be safe. I would say Korea is a little bit more strict into that, but again, it depends on the interpretation. And if you want to go to Korea, we advise to take a local expert when there is specific regulation that would let us think that it is not possible to use live microorganisms in cosmetic. But again, I think it's more about getting the right expertise locally. So now I think the most important part that would be interesting is the borderline. As I told you, I think why is it very important, the borderline aspect, is because when you're an inspector, this is the first thing you will see. And this is the most important to enforce. What is, before doing any testing, before even looking at the list of ingredients, is the cosmetic product wrongly presented as a drug? And I can see that by picking a product on the shelf or by just looking at its packaging. That's why at the moment there is a few enforcement with medicinal claims, and it's not just on microbiome, but any kind of medicinal claim. So if we look at the borderline challenges, we can have it from two sides. First of all, the application side. So we know that this product, so if we take a moisturizer, will be applied to the external part of the body, but is not going to have an effect so much on the external part of the body. It will have an effect, of course, but it will also have an effect on the microbiota that is present at this application site. So here again, there was some discussion about, well, do we fit the external part of the human body? I think, again, as long as it is presented as a cosmetic and it is applied externally, we have some argument in terms of the regulation to say, yes, it fits the application site requirement. And then we have the keeping in good condition, you see, in terms of the function of a product. The cosmetic product is not just about changing the appearance, it's also keeping the external part of the body in good condition. And here I would say, as long as there is no claims going to say it led to a significant impact on the body physiological function, such as changing totally the the balance of the skin, or treating an adverse condition, there is a way to be able to still put a product with microbiome claims in a cosmetic function to maintain the healthy status of the skin, to keep it in good condition, so that it's not going damaged by the environment, by a lot of things. So I think here that we can see that there is a way to classify such product as a cosmetic. The issue is that maybe what we are seeing is that, of course, we are seeing therapeutic claims on the market. We need to be a bit careful with that. And I think, as Katerina mentioned, it's very important to make sure that the claims doesn't overstate preventing or treating a disease. So here we had a lot of discussion about, well, can we talk about redness? Well, again, it depends why there is redness on the skin. Is it linked to a normal friction of the skin, or is it linked to rosacea, for instance? Atopic dermatitis, again, in the UK, when you refer to eczema, psoriasis, or these sort of things, you can say suitable for at a maximum. So there is a specific sentence from the authority that say that you can use, but the meaning is important. It's not just a sentence. What the authority would say is that a product that has a cosmetic function, but is also suitable for someone that has eczema, which means that it's not going to make the eczema worse, but it's not going to treat either, will still be as a cosmetic. So I think here again, I think it will be important to focus on the moisturization primary function, and maybe just if you have to refer to some of the claims, like the eczema or the psoriasis or the atopic dermatitis, to make that more like a, I would say, like a food claim. You see, when you are when you have a food allergy and they say, well, it's suitable also for people that have a gluten allergy, that doesn't mean that the product will treat or prevent gluten allergy sufferers. So you need really to think about that and be extremely careful. Some countries like the US will be even more strict with the word eczema. So again, I would say just be careful with what you are claiming for. And then once you are sure it's a cosmetic, claim substantiation, again, no specific rule for microbiome claims. You have to go via the normal regulatory landscape here with horizontal legislation, such as the Fair Cosmetic Commercial Practice Directive, the Cosmetic Product Regulation, some of the guidelines, and self-regulation. And here I say very importantly, with the ISA in the UK that is asking for robust evidence. So the key principle in the claims, I would say first, once you're sure it's a cosmetic, so making sure you are not making any medicinal claims. And sometimes I ask, can we use the word treatment? Can we use the word healing? Yes, it could be possible. I will never say no to that, but it depends on the context. You see a night treatment for a moisturizing cream that is clearly presented as a cosmetic, I will accept the word treatment, but something that is as a word treatment and referring to the acne or eczema, I will have a problem with that. So once you have eliminated the borderline aspect, then there is the efficacy of the product. You need to understand what has been claimed. And this is again what Katerina was saying. What does the consumer understand about the claim? The claims are very confusing. Are they very understanding this microbiome concept? Are they understanding prebiotic, probiotic, postbiotic? So again, trying to understand that. And once you have data on that, do you have sufficient data to support the claims you are making? And again, making sure you have it before you place the product on the market. So I would say that in the UK, we are very strict in the level of data that is needed. I think everything that will be on an advertising, such as TV, media, including digital advertising, is under remit of the ISA, the Advertising Standard Agency. And they will expect a very robust dossier if they ask you to prove your claims. And their view at the moment, and again, this will evolve with the science behind, is that there is very few microbiome claims that will have the supporting data. At the moment, they are happy maybe with some microbiome-friendly claim, but more than that, they will expect a big dossier. They are not saying it is banned, but they are just saying they haven't seen enough data at the moment to be confident enough that such claims will be supported. So I would say if you want to develop this sort of claim that are a little bit more on efficacy basis, maybe you want to make sure you have the right science behind that. Huh? And I would say the problem as well, in addition to the science, are the fact that, and this was mentioned by Kristin, is that there is differences in microbial composition between benchmark of skin. So the identification of the role these different plays, depending on whether it's a young person, an old person. You need to define the way how to influence the microbial composition. Is there really an improvement? And is it really interfering with the skin microflora? So I would say in a claim substantiation area, you have a lot of variability that you will certainly be asked by the ISA to prove that the data you have collected apply to the average consumer. And again, science is not impossible, but it will require a lot of work. You see, just having, I would say, some raw material data may not be sometimes sufficient. You will need to have this raw material data plus data on the finished goods and being able to justify that. So I would say, yes, we can put efficacy claim on skin microflora as a secondary claim, non-medicinal, but we need rigorous substantiation. Using the word help always is safer. I would say it's less as a category claims, but again, it depends on the type of claim that you are making. And as I say, one that is maybe more acceptable at the moment is, but we still require data. I'm not saying you don't need data, is microbiome friendly type of claims. And as Kristin and Katerina mentioned, we don't have a particular definition of microbiome friendly. So there is some sort of logos that are being developed, standards are being developed that could be useful for you when you develop a product to follow their standard, or at least to have a scientific reflection in-house. Huh? So that's all for me.
So thank you very much, Olivia. We have a couple of questions. I know you need to leave us. Do you have a moment?
Yes, of course. Yeah. No problem.
Okay, so let me just see. First one was more or less a comment from Dr. Jeff Briggs from Boots. I think there needs to be a clear differentiation between ingredients which affect the skin microbiome for a benefit and bacterial ingredients with a skin benefit. You don't need a probiotic to affect the microbiome. Consider Kristin's microbiome-friendly process. And the probiotic might have a skin benefit without having any direct effect on the skin microbiome. So I'll just very briefly invite you to comment, please.
Yeah, no, I agree. I think the formulation aspect and will be different depending on the ingredient. And I think you see from a perspective on the regulation part, it's more about developing some criteria at one stage. And I would say you just need to be careful. I would say the thing that we need to be careful is the transition of the science behind and all of this concept, and how you're going to communicate that to the consumer.
Lovely. And one more question from Vidu. Considering cosmetics are not for treatment, how does claims such as anti-dandruff that would target dandruff associated with CCUK, for example, would fit in?
So I think for the anti-dandruff, again, you see, this is something, if we look at the UK and majority of the European country, they will accept that as a secondary claim. So a shampoo, a conditioner with an anti-dandruff benefit will be acceptable. So that means my primary function is really to wash my hair, and at the same time, I have an anti-dandruff benefit. So we say, don't go crazy on the claim, like treating dandruff and as a proper treatment that will be seen as an over-the-counter. But yeah, anything that will be linked to a first cosmetic function will be acceptable. And this is why we are seeing a lot of shampoo, anti-dandruff shampoo, conditioner, anti-dandruff conditioner that are on the market. Huh?
Thank you very much, Olivia. I know we need to go now, but we will note all the other questions and share them with you.
Super, thank you. Bye-bye now.
And Sudeep, I will now invite you, I've made you a co-host. I'll invite you to share your presentation, please.
All right, that's great. So good morning, good evening, good afternoon, everybody. I'm gonna try and get my presentation up here. Hopefully it'll give me a heads up when you're able to see it.
We can see it now.
All right, fantastic. So my apologies, I was not able to join earlier. I know there was some change in schedules and I know we're sort of running a little over time here, so I'll try to breeze through this. And title my talk, Microbiome Growth Opportunities. And I'm Sudeep Basu. I serve as the Global Practice Leader for Innovation Services at Frost & Sullivan, which is a global growth consulting company. And I am grateful to the National Biofilms Innovation Center, as well as the Cosmetics Cluster of the United Kingdom, for having extended its invitation to me. So a little bit about myself. You know, in a previous life, I did have a PhD in microbiology and immunology. So earlier, whenever somebody would ask me, what's your PhD in? I would say microbiology and immunology very loudly. But these days, I tend to say microbiology louder. So that tells you a little bit about the shift that's taking place and the dynamics, especially on the commercial as well as R&D side. Some of the organizations I'm associated with, I serve as guest faculty at Rutgers University, New Jersey, and I also lectured occasionally at Stanford and a couple other universities around the world. I'm a reviewer for the NIH, which is the National Institutes of Health, the National Science Foundation, as well as the Federal Labs Consortium, on commercialization grants and/or technology transfer grants. And I serve as an advisor to a couple of universities on the commercialization side for med devices as well as pharma life sciences. And recently, I've started serving on the cell and gene therapy regenerative medicine standards bodies. And Olivia actually showed you some material about the International Standard Organization standards followed in Canada for some of these applications in the skin microbiome that she was talking about. I serve as a voting member for US TAG to ISO 2C76, which is a biotech run by NIST. With that, this is our global client network. I'm hoping some of our clients are on this call listening in, so you can recognize yourself. Thank you for your partnership. And start right in about some of the growth trends. And you know, if you can see the patent publication growth trends, that curve just keeps going up and up and up and beyond 2017 up to 2020. Haven't really seen what's going on in 2020. So maybe we should look at what the impact of COVID has been. I would imagine it would have been, you know, sort of somewhat flattened. But the microbiome venture funding scene is really good. Going into skin, because the discussion is sort of more specific about skin, you know, you could see that the skin microbiome related sort of patents that were analyzed by my team here, great work done by Manmohan Madhurya, Dr. Cecilia, and Vandana. And they looked at, you know, what are the different applications. So cosmetics was 23% of the patent portfolio. Personal care next. I don't know why Chinese SMEs are there, but there's a lot of utility patenting out of China. Biopharma, ingredient suppliers. You know, we were talking about ingredients just a little while back about their interaction and impact on the microbiome, as well as the biofilm formation, quorum sensing, things of that nature. Patent trolls, they get right in the middle. Those guys, you can't escape them. And then you've got financial institutions and so on and so forth. So if you look at the key players here, you could see under corporates, you've got L'Oreal, you've got Gojo, P&G, Cell Biotech, Henkel, Unilever. Several of those are our partners. You look at the research institutes, you got right from Harvard, Ohio State, UCal, right? Korea Institute of Science and Technology, KIST, interesting one, remember that, we'll come back to it. Of course, the Japanese guys. So it seems like there's quite a preponderance of Japanese and South Korean research institutes. And of course, in the startup landscape, you've got Biofilm from the US, you've got Biomedic from Europe, and then you've got several others from the rest of the world. And that's sort of a lay of the land of what's going on in terms of startups, et cetera, research, corporates, and functions. But I'll tell you this, that the interest in microbiome has become so ubiquitous that I'm getting to hear from people that are in pharma biotech, that are in food, and that have been in sort of probiotics. I remember when I was an undergraduate getting trained in microbiology in India, I actually had got a book out of the library on probiotics. It was this 400-page hardbound black covered book, and it's like, what the heck are probiotics? And that was my introduction. That was way back when, but the field has certainly evolved. And if you look at the key countries where filings are taking place in terms of key geographies of market protection, as well as by where the origin of science is, you'll see, of course, the US, China, South Korea, WIPO and EPO, of course, through the PCT, but Australia, France, Japan, Brazil, and Germany. But South Korea is really surprising. It's kind of up there, right? So you're expecting them to do great stuff in electronics. They're obviously doing great in energy storage. Now, of course, in the microbiome and healthcare side as well, they're coming up. So that tells you about the gradual maturation curve of a nation by looking at what kind of work they're doing in different research areas, just some of the high level trends we look at. Specifically in terms of emerging research trends, in terms of skin and hair, you could see the split is about 35% for skin, 1% for home health, a little bit for hair or scalp, and then the rest is oral in terms of just these segments, if you look at just these segments, right? And an FMCG, CPG players out there, if you're listening in, you could gain a major competitive advantage as a leading consumer care brand by acquiring or developing a differentiated skin or oral care offering with certain technologies, right? So happy to chat more about that. In terms of emerging trends, you could see the research studies or publication data. Those towers tell their own story. It's sort of an upswing field and pretty much everywhere, but definitely for skin, hair, and scalp, you're just kind of seeing a little bit of up and down. With regards to the landscape of microbiome, I don't want you to go away from this talk thinking that microbiome only impacts the skin. Obviously, it has much broader impact across areas, across GI. There is, of course, impact on the gut-brain axis, which is your neurological symptoms. So diseases like Parkinson's and Alzheimer's also have certain roots in gut bacteria, kind of work that's been done by Sarkis Mazmanian's lab, as well as my friend Dr. Friedland's lab, have both shown in papers published in Cell and Nature reports how the gut-brain axis can actually lead to disease causation, especially for PD and Alzheimer's, including some aggregation of fibers, et cetera. Food and beverages, we've kind of heard about that from previous speakers, I'm guessing. Personal care, we're talking about that, but also indoor air quality, right? So another interesting area that is starting to get impacted by the microbiome technologies is sort of the heating, ventilation, and air conditioning, what we call HVAC here in the US, and looking at reintroduction of probiotics in an indoor air environment. Now, because of COVID, you've heard a lot about HEPA filters and how a HEPA filter on a plane will completely protect you from the person seated next to you who's sneezing and coughing out a nanoparticle-sized virus. Absolutely, it will. But beyond that, what it also is doing is it's taking away some normal, flora that was perhaps in the atmosphere. And they're looking at reseeding some of that. If you've got allergy season, is there something else you could do there? One of the effects that hasn't been talked about much is the germ-free hypothesis, right? So because in more and more environments, you're getting lower and lower microbial loads, your body's immune system actually doesn't have much to react to. But we were not geared like that. For thousands of years, our immune system and our biology has evolved to actually fight pathogens, to fight viruses, to fight fungi, to fight microbes, not get totally purified water, food, and air and contact with surfaces, right? So imagine now, because of COVID, what's happening is we're completely sanitizing an environment, and we should. But one of the side effects which will happen is our immune systems will kick into overgear, not for the ones that are getting sick, but the ones that are actually staying healthy by staying away from the immune sort of antigens. And you could imagine what the role of microbiome will be going forward in sort of moderating some of those immune responses. I encourage you to think about that. Diseases like cancer, diseases like autoimmune responses could actually see allergies, could actually see a heightened sort of occurrence going forward in the coming years, especially kids that were born in this period that have been completely screened from a microbial environment. So it's probably still good to get those kids out somewhere to get some microbial interaction, but of course, stay away from the virus. Jumping gears into microbiome technologies that are transforming skincare. So what are the key drivers, right, for microbiome, apart from researchers just wanting grants to run their labs? I say that half jokingly. So rapid advances in microbiome research, are occurring across areas like biomarkers and bioinformatics. Those are big platform tools. Obviously, if you're looking at RNA-based qualification or sequencing to identify what strains are going in where, collaborations are rising on the microbiome front, right? If you're talking about companies like Procter & Gamble, Unilever, or any of the pharma companies, even manufacturers like Lonza, that actually do biomanufacturing and are completely preoccupied right now with vaccine manufacturing, are coming up with new protocols, methods, centers, and capacities and capabilities, right, for microbial manufacture as well, because you got to be able to do it at scale if you really have to take this to market. And those are companies that are looking at it. Increased demand for free-form ingredients. We were having a lively chat about ingredients and their impact on bacteria a while ago. Looking at consumer awareness. So, you have everybody jump in to go to Whole Foods and say, I want my probiotic. In fact, I had my mother visiting us from India some time back, and she said, I'm not feeling well. I think you should get me a probiotic pill. So what? But that's how commonplace this is, right? Now, and the growing demand for personalization, of course, we talk about precision medicine and personal medicine. Those are also the levers. What are some of the challenges, right? So the clinical validation of microbiome research, I think getting the data, getting the science right, making sure you're drawing the right correlation, that is important. And I think the onus is even more now on the scientists than before, because earlier you could just find a fact and say, oh, maybe it works like this. But now you really have to understand the mechanisms, because you're already going into drug development and you're going into regulatory challenges and so on and so forth, as we heard. Also, limited availability of shelf-stable probiotic strains, and I would say also R&D data on strains. Defining the global regulatory guidelines and pathways. I know we heard a lot about that in terms of the diverse landscape and what different countries are doing. We've done considerable amount of work on testing, non-animal testing using 3D chips, organ on a chip, cell on a chip, sort of testing system on a chip kind of contraptions that countries are now talking about, because you don't want to have cruelty to animals, no skin testing on animals, etc. Achieving optimal scalability we just talked about the Lonza example and getting in the game early for biomanufacturing, biomonitoring as well in terms of are there contaminants in the culture, just the usual stuff that you would do for any kind of scale up of biomanufacturing. And then on the business side, sort of production costs, regulation could be one of the challenges as well. So I don't know if that image is familiar, but we were looking at certain segments in terms of where is the skincare play happening. So there's probiotics, probiotic-derived products, again, something that was alluded to in the discussion. There's prebiotics, targeted antimicrobials, and microbiome-friendly ingredients. We can come back to this in a little bit if I have time. I really want to check on something here very quickly. Okay, all right, I probably have another 5 minutes. Okay. So AOBiome's platform, very interesting patented proprietary technology for intranasal formulation, incorporating a strain for beneficial AOB, that's Nitrosomus eutropha. And AOBiome has gone through with their trial, phase 1B for pediatric applications, but they're also doing some for adults. And I think those are in like phase 2A or others for eczema, which was the latest data on them. That was a US-based startup out of Cambridge, Mass. From Belgium, S-Biomedic, founded with Horizon 2020 program funding. I think the Horizon 2020 program has been one of those amazing programs that has done fantastic work for the European Union. I have myself visited several centers that benefited from the FP7 funding in Europe, especially in Poland, also in Brussels and Mol and Antwerp. And the kind of impetus that the Horizon 2020 program has given to European science is phenomenal. And not just science, commercialization, technology building, technology scaling, all of that, intellectual property portfolio creation. I think countries like the US have to really wake up and look at the success of that program and think hard about what we're doing here with regards to how we're looking at science, how we're looking at science-based decision-making, how we're looking at investing in the future of R&D here at home in the US. So this company actually, I'm going to skip through some of the details. I don't like all these text-heavy slides. You can see what are the different indications that they're working on. So acne, aging, dandruff. So you could see, skin, general health, and senescence, as well as hair. And then some of the partnerships, right? So they were part of the J Labs, which is the Johnson & Johnson labs. They've got a partnership with DSM as an investor from the material side. That's a big player. DSM is huge into the greening of materials. And sort of that's how they got started. And of course, European EU Horizon 2020 funding, a partnership with Flanders Bio and PRI on the regulatory side. But we're not just focused on the US and Europe. We're also looking at Asia Pacific in a big way. And when I say we, I mean Frost & Sullivan and my team that comprises of life science experts in microbiome specialists, et cetera, on the business as well as R&D side, really. And we're looking at countries like Australia. There's some companies there, China, of course, Japan. We talked about South Korea, Malaysia, Singapore, pretty much all over the place, really in Asia that has some interesting activity there. And you could see the companies, key players from each of those regions that are active. Now, an interesting anecdotal sort of piece of information that was shared with me by the team, and I thought this will be interesting. So it's not only humans that have racial inclinations, even bacteria do, okay? So absolute joke. So you could see that based on our melanin content and pigmentation and genetics, of course, skin color changes. And, with that, the chemistry changes, the biochemistry changes. So of course, the microbial colonization changes. So the ecology, the microecology, the niche environment changes. So that's not a surprise really for me as a microbiologist, but it makes an interesting, colorful chart to share. So ethnicity impacts microbial composition, but that goes back to another important part, is the personalization aspect. So maybe it is not just personalization to the individual, but maybe there is certain segmentation that you could talk about based on genetic makeup, genetic profiling, and skin type profiling. And companies like L'Oreal have done some significant amount of work there where they've tried to look at not just physical chemical properties, but other tests that they could do to categorize their users and end users, and then develop stuff for segments of those users, right? So this is about advances in technologies for skincare. I'll quickly flip through these. You can see some of the companies, Galani, Biofilm, SEUN, Zitra. Then you've got Estée Lauder, Clinique, so on and so forth in different areas. Naked Biome, So the role of the skin microbiome sort of in dysbiosis or conditions such as acne has been well proven. But I think the critical part is also the interaction between skin, the epithelial surface, the nasal mucosa, the brain nasal mucosa axis, the oral cavity, and the presence of bacteria there, the types that are there, as well as the gut-brain axis. I think they're all connected in different ways, and those connections have to be explored and further analyzed. I think going back to the science is one of the challenges, getting better with this, and it's happening. In terms of the market segmentation predictions and disease areas, so we base 2020 to 2025, look at cancer, right? I mean, that thing has just tremendously grown. And I know there are companies working in that area quite a bit. I would also encourage you to think in terms of the immunological connection between inflammation, cancer, presence or absence of certain types of microbes and what they do in the body and the metabolic impact that they have. But that is sort of a disease segmentation. And then approach to commercialization across the microbiome space, right? So how does an early stage company play this game? If you take the example of Biofilm, they do both, right? You get to the OTC first, and then you kind of build up the longer term plays on the therapeutic side, secure more funding, maybe you do some diagnostic work or some target profiling work, and maybe research spin-offs, what have you. And then again, that is my contact information, and thank you for inviting me.
Thank you very much, Sudeep. Really, really interesting talk. So I have some questions. I would like to thank all the speakers now, but I will open the Q&A session now. So I have Steve, could you stop sharing, please?
Yeah, sorry, go ahead. I'm going to do that.
So Kristin, if you can unmute yourself, please, and Sudeep, and it will be myself, Olivia had to leave. So Steve Barton, consultant to the personal care industry, is asking me, so that is probably for Kristin primarily, is the term microbiome friendly the best way of conveying what we are trying to communicate? Given that we still do not know, will it be a hostage to fortune? And is everything that is microbiome friendly, skin friendly, whatever that means? So I'll just copy this for everybody to read. I think that's easier. Let me see. So again, and the reference I had to Tony Rowling saying the dynamics of the exfoliation is vital. So the question to you, Kristin, is the residual core microbiome, the deeper one, able to cope with that, providing that the skin is healthy and not compromised by other factors? And other questions, what is the key determinant of friendliness, diversity, recovery, skin impact? And we know from experience that terms like these grow a life of their own, with different groups taking their own meaning. And you've demonstrated that not everything is substantiated by science on the market. So that can create consumer confusion. So what do you think?
So just for the first question, is the term microbiome the best way of conveying that we can, what we are trying to communicate? So I think that's what we are saying. If you're looking at healthy skin, it's not at the moment, so from what we know, it's not possible to do anything to the microbiome that would make it healthier on the skin. But of course, if you look at conditions such as acne or psoriasis or atopic dermatitis, you have specific bacteria that you know that are highly abundant, and you can kind of reduce them if it's possible. So it has been shown, for example, that some probiotics, live probiotics, would, but also I think even not live probiotics, have shown to milder symptoms for eczema, for example. But for healthy skin, I would say the only thing you can do at the moment is to not disturb the microbiome.
Thank you.
And then I just to go through the question again, is everything that is microbiome friendly skin friendly? Okay, that is a good question actually, because of course there's other points that can of course irritate the skin, which is not affecting the microbiome. So that is not given. So our seal is not saying if anything is skin friendly, but I think that is something what cosmetics usually have to go through when going on the market, right? So they have to test if there's any skin irritation in other tests. So we are only looking at the microbiome, but of course that is not, it's not said that it's skin friendly if it's microbiome friendly, but of course it's it's on top of skin friendly. And
I think then so I guess overarching question is, should we focus on the skin benefit after all?
Yes, that is something that has to be considered when like if you look at the cosmetic, you have to test both, of course, like not only microbiome but also the effects on the skin.
Mm-hmm. And what is the key determinant of friendliness? Diversity, recovery, or skin impact?
I would rather say, so it's, I'm always saying that diversity is not the key, but it's the balance, because in some cases, the higher diversity goes with symptoms rather than being healthy. So also if you look at the vaginal tract, it's rather a very distinct proportion of bacteria living there, and the higher the diversity, the more pathogenic it is. So it's rather, I would say, the balance and the balancing of the diversity we have on our different key on our different environments on our body. So we want to keep this balance intact and not disturb it in any way. So I would say that is key to health. And that will also reflect on this healthy skin then. Okay.
So perhaps just the last part, and then I'll go to what Jill is saying down there. So we know from experience that terms like these grow a life of their own, different groups taking their own meaning. This can create consumer confusion. Could you comment on that, please?
So so where is the question? Can you, is it written?
Yeah, so overall on the chat, if sometimes it's hard for the consumer to understand the detail of all this. How best to prevent consumers being confused?
I mean, yeah, that is on one hand, it is important to educate. It's like every brand that has something to say about the microbiome or says it's a microbiome kind of good for the microbiome. It should show the data on their, at least on their website. And I think it's important that the customer becomes educated about the topic. And that is also something that we're doing with our website, that they know what we're actually talking about when we're talking about the microbiome. And so then they can, and I think one has to be very transparent and show the data behind.
Thank you. Jill Westgate, is the diversity of microbial strains going to challenge mass product formulation?
Yes, so maybe mass product formulation is rather a problem because it's mass products are rather cheap and their ingredients are rather cheap. And we have done a lot of testing on mass products to see how our test performs. And we see that those mass products don't pass the test. So they are rather aggressive and have highly effective preservatives. And they kind of like, they have to smell good, but they have to also like have a long shelf life. And that's why they have a lot of preservatives and they will kill all of them, of our microbes.
Okay. What about commensals? Jacobo is asking.
You probably talk about probiotics, right? Like, or what?
I believe so.
Like apply commensals as a probiotic. That's what I also said, like it's getting dark outside. Maybe I should turn on the light. So that's what I said. We still don't know if we do something bad to our body if we add any commensal on top of our healthy skin. And that would again most probably lead to an imbalance again, and then lead to dysbiosis and to a skin condition that we don't want. So that is something that in the future might play a role, but we have to do a lot of research there to exclude downsides of it. And most of the commensals on the skin are not proven to be safe yet. So that's why probiotics for the skin are normally taken from gut probiotics that we know, because they're already proven to be safe in many, many studies. So.
Okay.
I had a comment, if I may.
Yeah, that's fine. We will go ahead.
Yeah, I was just going to say, if you're looking at profiling of microbiome for gut or for skin, you could potentially have curves where you're seeing the different populations of the species and strains of bacteria fluctuating across skin types or disease areas. And you could then normalize that and say, okay, if I'm seeing a particular commensal, or if I'm seeing one outlier, what is the impact on disrupting that skin microbiome ecosystem or the GI? And if that will be a sort of pathogenesis, right, for some disease type. And if that's a normal chart or profile, or is that a dysbiotic profile leading to some kind of disorder? And those kind of studies have been done for CD, especially in GI. I'm not sure, to my knowledge, I'm not aware if it's been done for skin microbiome. I would think that would yield some answers.
Thank you, Sudeep. We didn't see your face. That's why I was confused. So Jill is asking, there are many types of skin, so specificity will be key. So both Kristin, could you comment on this, please?
So yes, I think, so on one hand, it is important to not harm the microbiome, but if you're looking at specific skin conditions and you want to either promote the growth of a specific bacterium, or rather what is more likely at the moment, reduce the growth of a specific bacterium, then it would be great to have something specific that is specifically removing that pathogenic bacteria. And I know some technologies, of course, because that's the field that I'm coming from. And yeah, there's a lot of technologies there already, but they're not yet completely ready or not on the market yet. Some are, but yeah.
Can I just add that the industry develops products like body wash. You know, the body, as we see from those images, has maybe 25 to 30 different microbiomes associated with it. So do you think that the market for microbiome friendly and microbiome targeted skincare is going to become, you know, you use this on your big toe and that on your nose and that on your ear and another one on your underarms, and so that we're going to see a lot more body site specific targeting of products?
I think it's a question to both Sudeep and Kristin.
So yes, I think it's a good question. I mean, we already have kind of that. I don't think it makes a lot of sense at the moment to have like different products for every part of my face, for example. But still, if you want to address a specific condition, it would make sense to have it like cut for that specific area of the body.
Yeah, I think I agree with that. It also depends on what the market pull is for something like that. You know, what kind of market segmentation and growth you can still get by doing that. And if the consumer is going to view that. Now, specifically, if you're doing this for face, even from a cosmeceutical perspective, I think you might get a response. But if there's another disease or some pathology involved, which you're obviously, depending on the claims and everything, it would vary. At some point, I think in terms of it would be diminishing returns, even though scientifically you're absolutely right that is the way to go, because the flora changes depending on what the spot is and what the moisture content is and what the temperature is, etc. But from a market commercialization standpoint, there will be a few other factors, I guess, in the consumer's mind that'll drive uptake.
Okay. Sudeep, which sectors are driving research at the moment, please?
Yes, and thanks, Jill, for that question. Actually, I think it's pretty straightforward. If you see the data I presented, and I was actually looking at my slides, especially for skin, and I'm addressing only skin here, it is actually the cosmeceuticals and personal care that is driving it. Obviously, there are other types of diseases that you can address, but we have seen, at least in our work, that cosmetic companies have now become cosmeceutical companies, and they're really diving into what they could do to address these different disease areas. And I don't think that it's too far-fetched to say that some of these are going into pharma pretty soon, just the way they're developing these things. So the boundaries have really thinned out. So it's from the commercial side, it's those guys. From an R&D side, I think it's research across the whole microbiome space.
Thank you. Kristin, a question from Rainer. If all mass market products using preservatives kill the skin microbiome, why do so many human subjects still have a microbiota? Isn't it rather a question of the concentration, and isn't a bit too general to say that preservatives kill the microbiota on the skin?
Yes, I know. I remember, Rainer, your presentation you gave on the last conference. And you're right. It's not that you're completely like washing yourself and killing everything off, but you have your core microbiome, and the microbiota will come back on the surface. And of course, you're not completely killing off everything by washing just once. But like by repeatedly washing and using different ingredients, depends on how much you're using, you will more and more lose your microbial diversity over time. And what we're actually doing is to at least say, okay, those products that we're testing, they're not going to completely like kill the bacteria in our reaction flasks, but of course they will not completely kill your microbiome in when showering. But I would recommend not to shower too much and not to use too many products, and keep it simple and not to add too many products to your skin.
And Kristin, question from me, follow up on that. With COVID, the amount of hand sanitizers we've been using and the amount of hand washing we've been doing, what do you think is sort of the long-term impact of that for doing that for about 1.5 years till the vaccines kick in? I mean, I can feel my hands have become super rough by doing this, and I tend to use a lot of warm or hot water, even though, you know.
So for me, it's kind of almost hurts me when I see people disinfecting their hands once they enter the supermarket, and then again when they leave the supermarket. So that is something that rather does harm to you than protecting you from any virus. And because I mean, most people don't even use it in the right way so that it would really kill the virus if it would be there. And I would recommend, we even published an article about that on our website, to not use disinfectants. And if you're outside the house and you're feeling like you've maybe touched something that might contain the virus, whatever, or it's not even just that virus, there's other pathogenic bacteria and everything around, and just try to not like touch anything in your face during being outside. And once you come home, just wash your hands with not too hot water, and that's way better than disinfecting it, because that's, I think, contraindicative, and it's even not recommended by the WHO or German institution, the RKI. Yeah, if you want, you can read that article on our website.
Sure, I would love to. Thank you.
Okay, there is a bad line there. So a question from Jeff and Holly. So what's the panel's view on how to study the microbiome in terms of the impact of a product? They are constantly impacting on our microbiome, washing hands many times a day, you said, showering, the biome, certainly in terms of the quantity of recoverable microbiome material, DNA, by washing, and takes time to recover. So how to study it, I think was the question.
It's that is a very good question, because that's also something that I'm thinking of a lot, and that's also how we developed our standard. Optimally, it would be nice to have a test on humans, but as we just discussed before, you don't see an immediate impact of the products on your skin, but rather it's a long-term effect. So you would have to, if you want to perform a study, to use one product over a long time and over like 2-3 times a day. And then on the other side, you also have to like remove all other products from your daily skincare routine. For example, if you think about UV protection, that it might also have an impact on your skin microbiome. So you don't want to leave that away in summer. So I think it's really, really hard to do good studies on the skin microbiome, because there's so many other impacts on your microbiome that it's hard to really connect it with that one product that you want to test.
Okay, thank you. So Sudeep has signed off. So one last question and perhaps 2 more comments. Holly is asking, how do you feel about educating the consumer when mixing products? And she mentions that Korean skincare regimens are growing in popularity rapidly, layering many products with specific active ingredients on top of each other, especially as the target consumer of these products would often be people with acne and psoriasis. Could you comment on that, please?
Yes, so that is something that I also don't like a lot. It's also like that in the US, but I think it's becoming better that women, especially women, feel like they have to put like 10 layers on their skin to look beautiful. And I think that's it's also kind of my personal opinion. That's not true, and we don't need that. Like, if you have a beautiful skin, you don't need to cover it with layers. And on top of that, as in this question is already saying, it's harmful for your skin. So it has so many different impacts on your microbiome and on your skin that you don't even know, and you cannot really test it. But and if you have a condition like acne or psoriasis, it's going to be even worse, because your skin is never able to recover by itself.
Thank you. So just a few comments. Steve Barton, too much showering will gradually destroy the barrier anyway, irrespective of the effect on a microbiome. So we focus on skin benefits, again, as suggested earlier, perhaps. And Jeff Rex from Boots says the same. So should it be the skin benefits after all we focus on?
So I think we have to always take it as an interplay between the skin and the microbiome. It's not only the microbiome and it's not only the skin. And for example, if you look at the showering and at showering hot, then you also you strip away your sebum, because and actually it's something that you want, because you don't want to have an oily skin. But then of course you take away the nutrients for your bacteria, and then it's a kind of a viscous cycle. And then afterwards, because your skin feels dry, you add some body lotion, and then you again have something that disturbs your microbiome, and your skin is adapting to that. Probably you all know that the more you put the lotion and cream on your body, the more the skin gets used to that, and it's not being able to re-moisturize it by its own. So the best guess and the easiest thing to do is always, and that is what I'm always saying, to use as little as possible on your skin.
Thank you, Kristin. And last question, I believe, by Jill. What is your experience of ex vivo and in vitro skin equivalent microbiome models? So perhaps I've shown that within our 52 universities, we have both the ex vivo and in vitro expertise, and they truly differ in terms. So you need to look at what you study, what host response you try to evoke. So both have pros and cons. Kristin, would you like to comment, please?
Yes, so the ex vivo is of course also something that is very interesting, but also here, as you said, it's depending on what you want to see. And ex vivo is also considering how the skin in combination with the microbiota would react on a certain product, and that is very interesting. But also here you have to take different setups into account, because here you also have to, kind of look at different skin areas again, as we also do, like not only have one set of bacteria, but different set of bacteria that you can test for different representations of skin. But and also the skin might be different, have a different physiology in the different areas. So it's really, the more you add to a experiment, the more complicated it gets, and you really have to know what you want to know, like what you want to find out.
Thank you. One more question, actually, Kristin, it's for you. On studying skin microbiome, you mentioned having different databases that can lead to different types of, and I believe it's conclusions. Is there any effort that is initiated to address this issue? Is there a way to have a standard database?
Yes, I think there's some, like the major microbiome researchers we all know, like Rob Knight and Martin Blaser, and like all the researchers around them. They try to have a standard testing procedure and a standard database, and like to have like all like worldwide, they want to do, or their goal is to create a standard methodology from beginning to the top that all researchers would follow. I think that's in work, but I'm not sure how far they have gotten until now, but yeah. That was something that would be ideal. So that would be perfect if everyone's following the same methods, and then we can compare, we can really compare the research to each other.
So again, on behalf of NBIC, I would like to concur with this, sharing our methodology, sharing our practices is a good way in looking into the future of research. Jill, would you like to say a few words about CCUK before I thank everyone, please?
Yes, I've got a few slides, but I'm not going to use them because I think we're all zoomed out. But I would just like to say that for those people that don't know us, the Cosmetics Cluster is an organisation that was set up just earlier this year, building on some work that we've done previously. And the aim is to bring together companies and researchers in a collaboration under different themes. And we've been working with NBIC and Katerina, looking at the area of biofilms in both oral care and skin, hoping to make connections between companies and research labs. So the Cosmetics Cluster, you can find us on the website, cosmeticsclusteruk.com. Anybody who's interested to get our newsletters, please go onto the website and sign up for the newsletter. And then we share news, we share information on events. We're part of the Global Cosmetic Clusters Network, which involves, I think, 13 clusters all around the world. And we can help with anybody who's interested in connecting with clusters in other countries, as well as Alistair Gardner on our team is part of UK Trade and Investment and can help with questions on importing and exporting. So if you have any questions, drop me a line or follow us on LinkedIn, on Twitter, etc. And it would be really nice to get some of you on our newsletter circulation. So go to the website and cosmeticsclusteruk.com. Okay, thank you. And thank you very much for all attending today. It's been really great. It's good to have a big audience for our speakers. And thank you very much to Kristin for fielding nearly all of the Q&A. You did a great job.
Thank you.
So thank you very much from me and Vic as well. I've just asked Natasha to put up our website. So Innovation Hub for partnering for academia and industry for biofilm research. Thank you all the speakers and the audience, but also I would like to thank the teams behind who made this possible. So Jill and Jane from CCUK and Natasha and Vicky and Mark from NBIC. So have a good evening, everybody. Any questions you might have, we are happy to answer, and the recording will be available eventually. So have a good evening, everybody, and thank you.